So the next method is called as a grafting one. Grafting in the local language we call as a कलम, जिसको हम लोग बोलते हैं कलम करना. In the grafting, it is the process in which transplanting of one part of the plant on the other plant to form a composite plant. We are using the two part of the plant and we are joining the two part of the plant. Okay, this is called as a grafting technique. It is a transplanting of one plant. We are transplanting this plant on this rooted plant, which is rooted inside the soil one. And we are combining the two plants and becoming the whole one plant, which is called as a composite plant. This is called as a grafting technique. Remember one thing, it is a quick multiplication technique and proper growth of the better variety with weak stem. Okay, proper growth होती है यहाँ पर, quick multiplication होता है. The next important thing, basically it goes into the weak stem. Okay, the plant which is having the weak stem, they can easily undergo the grafting one, and it is the quick multiplication method where the growth is occurring. तो grafting इस तरह की टेक्निक है जहाँ पे quick multiplication होता है, growth होती है, and the next thing is that ये weak stems में भी हम लोग क्या कर सकते हैं. The next important thing about the grafting is that, as I told earlier about the two plants are joined to one another, in which one plant is rooted inside the soil. That plant is called as a stalk plant. See here, this plant which is having the stem and the leaf, it is rooted inside the soil, and this plant is called as a stalk plant. And the plant which is joined on the upper side, that is the cutted plant, that plant is called as a scion one. Okay, and the plant which is rooted, it's called as a stalk plant. Both these are get joined. Basically the grafting plant or cutted plant is called as a scion one. जो cut कर रहे हैं हम लोग, that is called as a cutted plant or scion. And which is rooted inside the soil, that plant is called as a stalk plant. The stalk plant should contain the disease resistance. These plants should have the disease resistance one. And the next plant which is implanted on this stalk plant, which is called as a scion plant, it should contain the high quality of the flower and the vegetables plant. High quality of flowers. High quality of flower should be there. Flowers and seeds. तो सियॉन में क्या रहना चाहिए? हाई क्वालिटी ऑफ फ्लावर्स और सीड्स रहना चाहिए. बर्ड्स भी वहां पे रहने चाहिए. नेक्स्ट, स्टॉक प्लांट ऑफ डिसीज रेजिस्टेंस. जब न्यू प्लांट आ जाएगा, दैट न्यू प्लांट कंटेन द बोथ द क्वालिटी, दैट इज द डिसीज रेजिस्टेंट एंड द हाई क्वालिटी ऑफ द flower one. So it is we are combining the two plants for the better quality of the new plant. The next important thing about the grafting is that the two stems, we are using basically the two stems. Here the two stems are get joined. The joining happen with the help of the cambium one. So the cambium of stalk is joined with the cambium of scion one. Cambium basically it is the layer around the vascular bundles. As we know that the vascular bundles are xylem and phloem. So this is nothing but the cambium ring is present around that one which joins the xylem and phloem. This cambium ring, where stalk of the cambium and stalk of cambium ring of scion will join together. They form a one common part. Okay, when these two cambium rings of stalk and scions are joining to one another, that is called as an organic connection. So organic connection happens in the stalk and scions. Okay, of the stalk and scions to develop a new plant. इस तरह से क्या होते हैं, दो stem stalk और scions के join होते हैं via the organic connections. Organic connection basically helps in the transfer of solutes from this stalk to the scion one. Solutes क्या हो रहे हैं, एक दूसरे में पास हो रहे हैं. Minerals are passing, and another organic food material, water is transporting from stalk to the scions via this organic connection or via this cambium ring. So this is the basic thing about the grafting. Grafting is of different type. Okay, as I have drawn over here, the grafting is basically divided into the 5 types. The first type is called as the tongue or whip kind of a grafting. In tongue and whip kind of a grafting, both the stalk and scion is having the same diameter. दोनों का diameter क्या होना चाहिए? Same होना चाहिए. Same edges will be multiple. Wedges are there. Wedges मतलब जिसको हम लोग बोलते हैं cut. तो दोनों में सेम साइज से कट होना चाहिए. दोनों का जो डायमीटर है सियॉन और स्टॉक के डायमीटर क्या रहना चाहिए? सेम रहना चाहिए. डायमीटर यहाँ पे हम लोग क्या यूज कर रहे हैं? वी आर यूजिंग द स्टेम. सो द स्टेम डायमीटर should be same both in scion and stalk, which is called as a tongue one or whip kind of a grafting. The second grafting, which is called as a crown grafting. Okay, here the crown shaped stalk is there. Okay, see here the bark size of the crown is much more than the scion one, and the multiple wedges are obtained in the in the stalk one. Multiple wedge हम लोग क्या कर रहे हैं? स्टॉक में लगा रहे हैं. It is looking like a crown one. That's what is called as a crown grafting. This is the crown grafting, in which multiple wedges will be there. It is looking like a crown. Stalk is much more in diameter than the scion one. Third one is called as a wedge grafting. Basically, the wedge is nothing but called as a cut, and this wedge or cut is made only inside the scion one. Stalk is remained as it is, and the scion size is much more less than the stalk one. थोड़ा सा कम रहना चाहिए scion का size, and wedge cut is made inside the scion. And remember, only one wedge is made inside the scion. सिर्फ एक ही wedge जो है scion में cut रहनी चाहिए to get fitted inside the stalk one. This is called as a wedge grafting. Next is the side grafting. Next हम लोग देखेंगे about the side grafting. In the side grafting, again the stalk size, bark of the stalk size should be much more higher than the scion. Okay, so here the cut is made at the side of the stalk one. The scion is inserted deep inside the stalk by their lateral side, by their bark side. That's what is called as a side grafting. Basically, these kinds of a side grafting is obtained when we are combining the two plants. One plant is of tree one, another plant is having a very weak stem. So it is easily get fitted inside the thick bark of the tree. So scion is having the weak stem and stalk is having the very heavy kind of a stem is there. Tree और एक जो है वेल का हम लोग क्या करते हैं? कॉम्बिनेशन जब लेते हैं, this kind of a side grafting is obtained at that one. Next is called as a approach kind of a grafting. Here in the approach, we are approaching our scion towards the stalk one. We are approaching the scion towards the stalk one. Okay, डायरेक्टली ऊपर से हम लोग क्या कर रहे हैं? ऊपर से हम लोग scion को ऐड नहीं कर रहे हैं. We are adding the scion by the approaching towards the lateral side of the main stalk. This is called as a approach kind of a grafting. So techniques will be there. That is a whip, tongue whip, crown grafting, wedge grafting, side grafting, and approach kind of a grafting is there. So basically हम लोगों ने क्या देखा? ग्राफ्टिंग एक टेक्निक है जहां पे हम दो स्टेम को ज्वाइन कर रहे हैं and forming a new plant. Rapid multiplication and growth will be obtained. It is a quick and an easy method, and which is easily applicable in a weak stem. Here, the plant which is rooted is called as a stalk plant, and which is get cut and joined is called as a scion one. And both are joined via the cambium ring. And stalk plant should be of disease resistance, and scion is of high quality of the flower should be obtained. And union will be take place via the cambium of stalk and scion one. So this is the whole thing about the grafting. The next procedure is called as a budding one. The name suggests budding is nothing but the use of budding. Bud is the compact structure which is present on the plant, which later get developed into the shoot and the root, or either they get developed into the flower one. We have discussed in the 11th standard about the floral bud, axillary bud according to their positions, and the apical bud which are present at the apex or upper region. So these are nothing but the compact structures. Bud are the compact structures which are going to be developed into the root, shoot or the flower laterally. Next, the what happened in the budding? Budding is the same technique which is as that of the grafting is there. We earlier seen about the grafting. In the grafting, the two stems are get joined to one another via their stem technique. Here in the grafting one is rooted inside the soil, that is called as a stalk plant, and which is cut from the other part and rooted inside the stalk, it is called as a scion one. Okay, so stalk and the scion is there in the budding one, but here in the budding the scion contain at least one bud, but the stalk is not having any bud. So the condition is that in the budding the scion should contain at least one bud. Okay, the next thing about the budding technique. The budding technique जो है grafting जैसी ही same technique है, and this budding technique is easier and very economical. Multiple copies are obtained. In the grafting technique, as we observe that some of the organic solutions will not form. Organic solutions will not form in the grafting technique. But here, with the help of the bird, the organic solutions are easily get formed, and the bird is obtaining the nutrient from this point of attachment. So it is much more easier and the economical technique. So this can overcome the disadvantages of the grafting one. The second most important thing, which is easily obtained in the rose, apple, peach and the sugar cane. As you observe that in the sugar cane, the long stem is there, and here at the internode some small birds are present at the sugar cane. If we dip this sugar cane again inside the soil, okay, if we are digging this sugar cane which is having the bird inside the soil, these birds will develop into the new plants. So we can obtain multiple copies of the same plant by digging technique. Another technique by grafting technique is there. So in the grafting technique, what is that? The stalk plant is there and the bird plant is there. So the scion plant is there. So the stalk plant, the bark is cut. Okay, as I told earlier that about the upper layer of the stalk is removed. Here the bark is exposed and deep cut is made inside the bark. एक deep cut हम लोग लगाते हैं bark में. Next, scion में मैंने condition क्या बताई थी? Scion should contain at least one bud. Again, the bark is made inside the scion and this bark should contain at least one bud. And it is at the needle shape. Cut is made in between the stalk and the scion. So they can fit easily with each other. But यहाँ पे किस तरह की fitting रहेगी? Scion and stalk should be made fit like lock and key model. Here these are covered by some clay material. They are joined and bandaged. हम क्या करेंगे? दो bark के contacts को join करेंगे, bandage करेंगे. But here remember one thing: bird should be exposed to the air. Later in the 2 to 3 weeks, what happens? These birds get sprouts. They develop into the adventitious shoots and roots. So these birds are again cut along with the stalk. यही bird जो sprouts हो गए हैं, stalk के साथ हम लोग क्या करेंगे? Cut करेंगे. And they are sown into another soil, and the multiple copies of the plants are get obtained. So this is the one of the technique of the budding. आप लोगों को ध्यान में क्या रखना है? स्टॉक में क्या नहीं चाहिए हम लोगों को? लीव्स और बर्ड्स नहीं चाहिए. ओनली बर्ड्स शुड बी देयर इन द scion वन. रूटेड प्लांट, स्टॉक प्लांट में हम लोगों को बर्ड्स नहीं चाहिए, लीव्स नहीं चाहिए, बट डीप कट चाहिए. नेक्स्ट, scion शुड कंटेन एटलीस्ट वन बर्ड. ओनली देयर बार्क आर सील्ड एंड द बर्ड आर एक्सपोज्ड टू द एयर वन. जब बर्ड स्प्राउट्स हो जाएंगे, कट दैट स्टॉक विद द बर्ड, along with the bird and rooted in another soil. So this is the technique. Are you video? So the last method is the micropropagation method. The fifth method, which is called as the micropropagation. As the name suggests, the micro is called as the micron in size. Micro means very small. Micron in size. Micron in size आप लोगों को पता है 10⁻⁻⁶. तो छोटा सा पार्ट हम लोग ले रहे हैं प्लांट का, उसको क्या कर रहे हैं? प्रोपोगेट कर रहे हैं. These small part, micron part, help in the propagation of the plant, that is the formation of new plant, that is called as the micropropagation. So micropropagation basically it is the advanced technology to propagate the plant, to form the new plants. And this one is, मैंने बताया कि this one is the advanced technology. And the rapid raising of new plants from the small plant tissues with the help of tissue culture technique. प्लांट के ही हम लोग क्या ले रहे हैं, स्मॉल टिश्यूज ले रहे हैं माइक्रोन साइज से, और इन्हीं स्मॉल टिश्यू से क्या कर रहे हैं, प्लांटलेट्स या न्यू प्लांट्स डेवलप कर रहे हैं. दैट इज कॉल्ड एज अ माइक्रो प्रोपोगेशन. Another definition of the micro propagation is formation of new plant with the help of micropropagules in the laboratory or in aseptic conditions. Aseptic means highly sterilized conditions is there. Micropropagules, these micropropagules, हम क्या कर रहे हैं, न्यू प्लांट फॉर्म कर रहे हैं with the help of micropropagules. Micropropagules may be the any part of the plant. It is a very small part of the plant which is having the buds, that is the adventitious shoot, root or leaf is present. That is called as a micropropagule, which helps in the propagation of the plant. ये फ्लोरल बर्ड्स भी रह सकते हैं, एडवेंटिशियस बर्ड्स भी रह सकते हैं, स्टेम के, शूट के, किसी भी टाइप के क्या रह सकते हैं, बर्ड्स रह सकते हैं, which will get further developed into the new plantlets. That is called as a micropropagules, but their size is about micron in nature. काफी स्मॉल साइज में हम लोग ले रहे हैं. That's why they are called as a micropropagules in laboratory or aseptic condition. So this one is a rapid method. हम लोग क्यों यूज कर रहे हैं माइक्रोप्रोपगेशन, हम लोगों ने डेफिनेशन देखी. नेक्स्ट हम लोग देखेंगे किस तरह से हम लोग माइक्रोप्रोपगेशन करते हैं. बिफोर दैट वन, व्हाई वी आर यूजिंग द माइक्रोप्रोपगेशन. इन सम प्लांट्स व्हाट हैपन, दे आर नॉट, जिस तरह से एग्जांपल लूंगी मैं ऑर्किड प्लांट का. ऑर्किड प्लांट basically these are the epiphytic plant, they grow on the trees, bark of the trees. Okay, so these kinds of orchid plant जो कि ornamental plants है, जो कि हम लोग यूज करते हैं, काफी महंगे प्लांट्स होते हैं. अगेन द साइकस बेदोमनी, दैट इज नथिंग बट द रेड वुड जिससे हम लोग हासिल करते हैं. दैट काइंड ऑफ अ प्लांट विल बी ग्रोइंग वेरी स्लोली, काफी स्लो ग्रो होते हैं. फॉर द रैपिड ग्रोथ, ओके, हम लोग क्या कर सकते हैं, अगर ये सीजन में ग्रो होते हैं, 3 या 4 साल में ग्रो होते हैं, इसकी हम लोगों को क्या मिलेगी, आगे जाकर जो है नए प्लांट जल्दी नहीं मिलेंगे. ओके, फॉर द फॉर्मेशन ऑफ द न्यू प्लांट्स अर्लियर देन द एक्चुअल साइज वी आर यूजिंग दिस माइक्रो प्रोपगेशन. एक्चुअल ईयर से पहले हम लोग क्या कर रहे हैं, यही माइक्रो प्रोपगेशन के थ्रू हम लोग प्लांट्स डेवलप कर रहे हैं. ओके, दैट इज नथिंग बट कॉल्ड एज अ माइक्रो प्रोपगेशन. कहाँ पे डेवलप कर रहे हैं, इन द लैबोरेटरी कंडीशन, दैट इज कॉल्ड एज अ aseptic condition. So the ornamental plant, the plant which are economically important, that can be easily multiplied within a short period of time. Okay, short period of time. So we can multiply. Okay, means basically economically important plants. Economically important plants are grown within a very short period of time under the aseptic condition. Aseptic conditions are necessary. Okay. The next is the, what are the actual procedure of this micro propagation. Okay. So what happened in the micro propagation? First, the plant which multiple copies has to be obtained, this plant is selected and select the part of the plant. Here I am using this leaf. From that part of the plant, the small part is get cut. Okay, that small part is cut from the meristematic tissues. Okay, means from the young growing part, we are cutting this part in a micron size, and this is taken as a X plant one. And that X plant is now get sterilized. हम लोग क्या करेंगे, पहले जिस प्लांट के हम लोगों को मल्टीपल कॉपीज बनाना है, उस प्लांट में छोटा सा पार्ट लेंगे. आफ्टर दैट वन, कट दैट. The micropropagation, as I told earlier, that it is the method in which multiple copies of the plants are obtained via the aseptic conditions. To choose the one plant whose multiple copies will be obtained, that is taken as a parent plant, and choose any part of the plant, okay, and cut that part. That cutted part is taken as a X plant. That cut part should be very small in quantity. It is about the micron in size. That part is now micron in size is called as a explant. Basically the explant contains the meristematic tissues. Explant में क्या चाहिए हम लोगों को? meristematic tissues चाहिए, because the meristematic tissues are having the rapid rate of cell division. Rapid cell division is there, so it can easily get multiplied in the aseptic or laboratory conditions. So take that meristematic tissues in the explant, and that explant was sterilized. It is surface sterilized by some techniques with the help of some chemicals, and that explant which is sterilized is transferred into the nutrient medium. Nutrient medium, basically it is a medium which provide nutrients to the developing explant. The explant is get developed inside the laboratory condition. Put this test tube in the laboratory conditions under the aseptic humid conditions. Light and temperature will be given according to the explant requirements, and this explant is now going to be converted into the callus one. So the callus is nothing but the unorganized mass of cell. Here lots of cells are present, that is the unorganized mass of cells. This callus is cut into the many pieces and transferred into the another nutrient medium. That nutrient medium is rich in auxin and cytokinins for the growth of root and shoot. So here, as you can observe, that the small plantlets each get developed inside this test tube. Okay, for developing of these roots and shoot, we need a proper amount of auxin and cytokinins. So this callus is now transferred into the another test tube which contains the proper amount of auxin and cytokinins for the development of root and shoots. And now another technique will be multiplied for the multiplication of these small plantlets. And now these plantlets, okay, these are get multiplied via the tissue culture technique. These plantlets are now grown in a greenhouse, and now they become the mature plant, and now they are ready to sell in the market one. So this is the method of the that is called as a micropropagation. From the small part of the plant, we are obtaining the multiple copies of the plants via the tissue culture technique, that is called as a micropropagation.